Bismillah ar-Rahman ar-Rahim. Welcome again to another important video. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University. Our video today will be about netosis in psoriasis. And actually, I already made 3 videos to describe in details the role of netosis in psoriasis. And this video will be the first one about the relation between netosis and psoriasis.
Netosis, as I have explained in another video, is a program of formation of neutrophil extracellular traps or nets that consists of modified chromatin which is decorated or filled with many bactericidal proteins that came from neutrophil granules and cytoplasm. And also, as I have explained before, the process of netosis needs reactive oxygen species. The main source of this reactive oxygen species is NADPH oxidase or NOx enzyme. And to activate this enzyme, we will need increase in calcium concentrations in the cytoplasm and in some cases generation of reactive oxygen species in the mitochondria. Basically, we have 2 types of netosis, the classical or suicidal netosis, which leads to cell death, and the second one is the vital netosis, in which neutrophils not only preserve its viability but also many of its effector functions. We have to know also that vital netosis takes only about 30 minutes. So it's very quick and rapid, while the suicidal netosis needs about 3 to 4 hours. Here is the main paper I took from it, the molecular mechanisms of netosis, which is published in Biochemistry 2020, and its title is Netosis, Molecular Mechanisms, Role in Physiology and Pathology. This diagram I took from the mentioned paper that shows that in the process of classical netosis, we need a NADPH oxidase enzyme to be activated. And to be activated, this enzyme needs pyruvate kinase C, which needs or could be activated by many ways, including increasing calcium, intracellular calcium. Here we can see calcium comes from endoplasmic reticulum, and then we get calcium from outside the cells. This calcium goes to the mitochondria, where it leads to formation of mitochondrial reactive oxygen species, and then this will result in activation of protein kinase C to activate NADPH oxidase, and this oxidative burst will lead to degranulation of neutrophil granules, and which will go to the nucleus, and then we get chromatin decondensation and formation of pores in the nucleus and in the cytoplasmic membrane to form the extracellular nets and complete the process of netosis. This is another paper talking about molecular mechanisms and therapeutic target of netosis in diseases, 2022. In this paper, it says that netosis is classified into 2 categories, as I said before: suicidal netosis, which occurs in the presence of NADPH oxidase or NOx activity, and vital netosis, which occurs without NADPH oxidase activity. Here is another diagram that shows the details of the 2 main processes of netosis, suicidal netosis and vital netosis. So in suicidal one, neutrophils become stimulated to start the process of netosis through different stimuli that will lead to endoplasmic reticulum release its calcium inside the cell, increasing calcium concentration, and this calcium will make activation of NADPH oxidase enzyme through activating its enzyme protein kinase C, and then we get the oxygen burst or formation of active oxygen species that will lead to degranulation of cytoplasmic granules and the release of its contents like myeloperoxidase and neutrophil elastase. And also increasing calcium concentration will activate PAT4. These PAT4 plus neutrophil enzymes will go inside the nucleus and make citronation of histones and decondensation of the chromatin, and then this chromatin covered or decorated with many enzymes like myeloperoxidase and neutrophil elastase and citronated histones will go through nuclear pores and then cell membrane pores to go outside and form the extracellular traps or nets for the formation or completing the formation of netosis. In the vital netosis, we have the neutrophil will be stimulated through different stimuli like complement or activated platelets, and then we get activation of something called SK3 that will lead to increasing intracellular calcium concentration, which will lead to activation of PAT4, and this PAT4 will go inside the nucleus to make citronation of histones and decondensation of the chromatin, and then we get the chromatin covered or decorated with citronated histones and also neutrophil enzymes like myeloperoxidase and neutrophil elastase. Then this chromatin will go to the cytoplasm, but inside vesicles, not free like the first one, the suicidal one. So we'll go inside vesicles, and then these vesicles will go outside the cell to complete the process of vital netosis. In this paper also they said that there's a special type of vital netosis which is called mitochondrial netosis. Mitochondrial netosis is dependent also on reactive oxygen species, and this one is contrary to the nuclear vital netosis, in which case the nets are formed from the mitochondrial DNA and not the nuclear DNA. And up till now, it's still unclear why the neutrophils choose to make netosis from the nuclear DNA or from the mitochondrial DNA. This could be dependent on the type or intensity of stimulating factor, but this process of choosing which one is not clear till now. This is a third paper which is published in International Journal of Molecular Science 2022, April. Its title is The Expanding Role of Extracellular Traps in Inflammation and Autoimmunity, the New Players in Casting Dark Webs. So they make an example like how much this netosis will affect or make a play in inflammation and autoimmunity, like players in dark webs. In this paper, they add to the two types we know about netosis. We know vital and societal netosis. They add a third type, which is caspase dependent, in which something called gasdermin D creates spores in the plasma membrane to allow DNA extrusion. So this third pathway of netosis or third type is caspase dependent. So to return to our subject today, that some researchers showed evidence of netosis involvement in classic psoriasis, but not pustular form. So there is some evidence that netosis is related to the pathogenesis of psoriasis, the classic one, and not the pustular one. So in this paper, for example, which is published in Frontiers Immunology 2022, in June 2022, they studied netosis in psoriatic arthritis by measuring myeloperoxidase DNA complex level in the blood, and showed that it correlates with the activity of psoriatic arthritis. So again, they found that there is increase in the serum myeloperoxidase DNA complex in patients with psoriasis, and this was correlated with psoriatic arthritis disease activity. And also they found that the more the active the disease, the better the correlation of serum myeloperoxidase DNA complexes level with the disease activity in psoriatic arthritis. So they conclude that netosis is involved in psoriatic inflammation, and serum myeloperoxidase DNA complex could be used or have the potential to be used as a useful biomarker to predict therapeutic response in patients with psoriatic arthritis. Actually, the presence of neutrophils is one of the known important histological features of psoriasis, with neutrophils located both in the dermis and within the epidermis, where they form small microabscesses in chronic plaque psoriasis, or larger intraepidermal neutrophilic collections characteristic of pustular psoriasis. It was an old observation that there is a pathogenic role of neutrophils in plaque psoriasis, that drug-induced granulocytosis improve psoriasis, while when we try to normalize neutrophil counts, disease exacerbation will happen. So if we try to make logic connections between the process of netosis and psoriasis pathogenesis, we can see that nets from one side have many proteases and enzymes that form main components of the nets, and these proteases and enzymes could make injury to the surrounding cells and augmenting inflammation. And from the other side also, extracellular traps or these nets release more than 70 known autoantigens that could stimulate the autoimmunity. And as we know, psoriasis pathogenesis forms a spectrum from autoimmune to autoinflammatory processes. So from the inflammation side, we are going to talk about the net T helper 17 axis in psoriasis and what happens with netosis, and also activating the keratinocytes by the crosstalk between toll-like receptor 4 and interleukin 36 receptor. From the other side, we will talk about the net responsible autoimmunity in psoriasis. So we will go to talk about the both sides, the inflammatory side relation and the autoimmune side, the relation between netosis and psoriasis pathogenesis.
I hope that you have enjoyed the video, and I'm reminding you again to subscribe to the channel and activating the alarming bell if you are interested in being updated by any new video. And thank you.